discuss about scanning electron microscope and transmission electron microscope. There is a general instruction about the electron microscope. The electron microscopes are instruments that use a beam of electrons to examine objects. Electron microscopes were developed due to the limitations of light microscopes. Another important feature in electron microscope is that electrons can able to mobilize from origin towards the sample under controlled environment, where it is absence of external environment. Like this tightly, completely compact setup is there. The vacuum is created within the electron microscope. So the electrons are floating in the vacuum towards the sample, and from the sample, the secondary electrons are released. They are directed by the detector in such a way the image of the sample is obtained. Electron microscopes are generally two types: scanning electron microscope and transmission electron microscope. Based on their working principle, it is named as scanning and transmission electron microscope. By using scanning electron microscope, the morphology or topography of the sample can be obtained, that is, surface or outer structure can be obtained by using scanning electron microscope. In transmission electron microscope, the electrons are diffused, transmitted through the samples, and internal structure can be viewed by using transmission electron microscope. The scanning electron microscope is a type of electron microscope that images a sample. By scanning it with high energy beam of electrons. In a scanning electron microscope, the secondary electrons produced by the specimen are detected to generate an image that contains topological features of the specimen. What are the characteristics can be viewed by using scanning electron microscope? Mainly topography, morphology, composition, and crystallographic information. Topology is about external features, that is, structure and material properties can be viewed. Morphology, the size and shape of the particles, and composition, what all the material is made up of, or the combination of the metals can be analyzed. Then crystallographic information, how the atoms are arranged in the objects. Construction of scanning electron microscope, it consists of an electron gun. It is a light source gun to produce high energy electron beam. A magnetic condensing lens is used to condense the electron beam, and a scanning coil is arranged in the beam. The scanning coil is arranged in the magnetic condensing lens and the sample. So, a scanning coil is arranged between magnetic condensing lens and the sample. The electron detector, it is technically called as a scintillator. It is used to collect the secondary electrons. It is emitted from the sample and can be converted into electrical signal. These signals can be fed into CRO. CRO is nothing but the cathode ray tube for viewing and photographic purposes. Here you can see the scanning coil is located between condenser lens and the sample. The electrons are passed from generated or produced by the electron gun. They are floating in the vacuum condition, and it hits the sample in such a way it is emitted from the sample. The emitted secondary electrons are collected or received by the detector. Such electrons are converted into signal by the photomultiplier. Then Such information is transferred to cathode ray tube, where the signals are converted into image in a 3D format. This is how the scanning electron microscope is working. It is the same one, the working of scanning electron microscope. How the sample can be prepared, because for every type of microscopic techniques, the sample preparation is must or important point. If we are not following or working carry out the procedure to prepare the sample, we may not get a good picture or good image. The specimen to be analyzed by electron microscopy has to be dry. The sample should be dry, with most biological samples are not, as dehydration might lead to structural changes. The specimens are first fixed to preserve their structural features. The sample should be dried during dehydration process or drying process. The structural information should not be changed. So it is essential or it is must that the sample should be preserved by using preservatives. The fixation is the first step and can be achieved using chemicals such as ultraldehyde. Generally, ultraldehyde is widely used as the fixating agent in not only the electron microscope and also in electrophoresis process. That is sodium fluoride sulfate SDS page preparation of the ultraldehyde is used to fix the protein in the gel itself. The fixed specimens are then dehydrated, usually by exposing them to an increasing gradient of ethanol. Ethanol helps to dehydrate the dehydration process. The dried specimens are then coated with a conducting material, usually gold, to make the surface conducting and cause it emit more secondary electrons. The dried sample is coated with a gold, which helps to emit more secondary electrons. Based on the obtained electrons or collected the information are converted by CRO. This is an example of scanning electron microscopic image of human erythrocyte, that is RBC. The advantages and disadvantages are it gives detailed 3D and topographical imaging and versatile information garnered from different directors. And the main disadvantages are it is expensive and large. Special training is required to operate such instrument, and very limited samples can be solid samples can be limited. A risk of radiation exposure associated with electron. The applications of secondary scanning electron microscopes are it is widely applied in virology, forensic science, anatomical picture studies, and characterization of nanoparticles. The second part of this presentation is transmission electron microscope. It was first developed by Ruska in 1930. A beam of electron is transmitted through an ultra-thin specimen. Here the sample should be ultra-thin because then only the electrons are diffused, transmitted through the samples. Whereas in the scanning electron microscope, it is not mandatory that the sample should be ultra-thin. An image is formed from interaction of the electrons transmitted through the specimen and detected by a sensor. The composition of the transmission electron microscopes are it's not much difference because here also they are the instrument is made up of or it consists of condensing lens. Then here the thing is in the scanning electron microscope the sample is at the bottom. That is after condensing lens the sample is placed, whereas in the transmission electron microscope the sample is placed between the condensing lens. When the electron gun, moreover it is nearby the electron gun, when the electrons are diffuses the sample and when it diffuses the transmitted electrons are collected by the receiver or detector, then the images are obtained. Here there is no photomultiplier or CRO. Directly The diffused electrons, here there is no secondary electrons or the electrons are emitted from the samples. It is not the case of transmission electron microscope because here the electrons are diffused through the sample and in such a way the images are obtained. The working of transmission electron microscope is stream of electrons are produced by the electron gun and is made to fall over the specimen using magnetic condensing lens. So condensing lens which helps to spreading of the electrons which are floated in the vacuum condition which throughout the sample, which helps to spread throughout the sample. Based on the angle of incidence, the beam is partially transmitted and partially diffracted. Here also based on the angle of indensing, that is angle of hitting of the electrons to the samples, they are transmitted as well as some of the electrons are diffracted. Both of these beams they combine to form the image. The combined images is called phase contrast image. So here the diffracted light as well as the transmitted electrons, diffracted electrons as well as the transmitted electrons both are combined together to construct the image. The combined image is called that image is called phase contrast image. The magnified image is recorded in fluorescent screen or CCD. I already told you that the sample preparation plays an important role in electron microscopy. The very first requirement of transmission electron microscopy is the sample preparation. The samples are usually fixed by the combination of ultraldehyde and paraformaldehyde. The second staining can be done with osmium tetroxide. Osmium tetroxide fixes the unsaturated lipids. So osmium tetroxide which fix the unsaturated lipids and being a heavy metal act as an electron stain too. Here the sample in the transmission electron scope, not like scanning electron microscope, where scanning electron microscope the sample is coated with gold, which helps more electrons, secondary electron productions, whereas in the transmission electron microscope the sample is ultra thin and it is fixed as well as the stained by using osmium tetroxide, which helps to fix the unsaturated lipids. The samples are then dehydrated. The dried samples are then sectioned to obtain ultra thin sections. The sections are then stained with heavy metal stains such as uranyl acetate and phosphotungstic acids. So after sectioning, the ultra thin sections are stained with heavy metal stains such as uranyl acetate and phosphotungstic acid. The stained sample is then deposited on a carbon coated grade. Here it is not, the sample is not coated with the coating material, but whereas the transmission electron scope, the sample placing grid is coated with a carbon and analyzed by transmission electron scope. This is an example of transmission electron microscope image of mitochondria. The advantages of transmission electron microscope is it offers very powerful magnification and resolution. It provides information on electron and compound structure. The images are high quality and detailed. So the disadvantages of transmission electron microscopes are large and very expensive, laborious sample preparation, operation and analysis require special training and maintenance cost is high like scanning electron microscope. Applications of transmission electron microscope is biopsies. In the case of biopsies, cancer result studies, toxicological studies and nanoparticle characterization studies are widely used. The difference between scanning electron microscope and transmission electron microscopes are electron beams passes through the thin sample in 10, whereas SEM, it is secondary electrons are diffracted by the sample, so it means that the electrons are not diffusers or transmitters. Specially prepared when samples are supported on temp trays. Sample can be any thickness and is mounted on an aluminum stud. Specimen stage or way down column. Specimen stage in the chamber at the bottom of the column. In scanning electron microscope, the sample is placed on the bottom of the column, whereas in the transmission electron microscope, the sample is placed between the condensing coil. The image is shown on fluorescent scheme, image is shown on TV monitor, image is two-dimensional projection of the sample, image is of the surface of the sample. That's all about electron microscope, that is transmission and scanning electron microscope. Thank you. This is Dr. M. Balasubramanian, Associate Professor, Department of Biotechnology. Thank you.